Thanks for joining us for the March 2025 iteration of the monthly seminar on physical genomics. My name is Benjamin Keene here at CPGE at Northwestern University. We're really happy to have our invited speaker today, Professor Lothar Schermelleh. He is Professor of Bioimaging and Academic Director of the Micron Bioimaging Facility at the University of Oxford's Department of Biochemistry. He's known for his pioneering work in the biological application of super-resolution structured illumination microscopy and the development of advanced live cell imaging methods, which have contributed to the understanding of nuclear organization and epigenome biology. Professor Schermelleh's biological research focuses on understanding how mammalian genome activity is related to 3D nuclear organization and on uncovering the interplay of biophysical forces, cohesion complex activity, and epigenetic memory for regulating cell type specific transcriptional programs during cell differentiation and in pathological states. So, Professor, thank you so much for sharing your work with us today. A couple quick notes for our audience. We'll do a question and answers at the end of the talk. So feel free to type them into the chat, either when you think of them or just wait till the end. You can also, turn your mic on and raise your little Zoom hand during the Q&A if you prefer to do it that way. If you'd like to check out more of our research here at CPGE at Northwestern, you can come visit us at physicalgenomics.northwestern.edu. We've also got an archive of these monthly seminar talks and other lectures on our YouTube channel, or you can follow us on Twitter X or Instagram. And one other note, we do have our annual seminar on physical genomics coming up in Chicago next month, April 25th. We've got a great set of lectures, our keynote speakers, Geeta Narlikar. So if you'd like to register, you can screenshot this screen if you like. There's a QR code there, also on our website or in the email blast you probably saw for this event. Feel free to register, submit a poster for the poster session, all that stuff. So April 25th. Anyway, that's my pitch. Professor Schermelleh, I think we can turn things over to you if you'd like to share your screen. And I can turn the floor over to you. Thank you once again.
So, do you see this?
I see you.
You see me? Oh, not too good. Let me see if I can change this. Share. There we go. Desktop 1.
There we go. Now we're seeing you.
And then, well, there we go. No, that's the wrong side of the screen. Feels like a throwback to COVID times.
We're all out of practice a little bit.
That went smoother. Let me see. How do I do this now? Here's the share thing. Let's try again. And then I'll just simply share the other thing. All right. Good.
There we go.
Okay,
Perfect.
I put this to the side. Now, well, first of all, thank you very much for the invitation. It's really a pleasure. I just talked to Ben, said it's been a really, really busy week and a long day, and I really hope I'm not completely running out of energy, but I give my best. So, yes, so our interest of research is really studying, organization of the nucleus and chromatin, and we do this by advanced imaging methods. And and if you look here at this kind of image here, of course, it's about pushing the resolution, the quality of our data from something like here in the corner to get a much clearer, more detailed picture. But what we're of course more interested is to analyze the data and get meaningful quantitative information that really informs us about the underlying biology, and then of course also use other techniques, imaging and other molecular images to cross-validate our findings. So for instance, electron microscopy, and of course not for this audience, I don't have to give a big introduction, but basically showing a classical textbook image of a mammalian cell nucleus with the classic annotation and different differentiation between essentially two compartments which is euchromatin, which is of course open chromatin, nucleosomes widely spaced, genes accessible for transcription, and heterochromatin, considered to be close, densely packed nucleosomes and inaccessible to regulators. Of course, when you then look on very small molecular scale, we typically draw chromatin as beads on a string that binds to transcription factor complexes that assemble at enhancers and promoters and exert their regulatory function to promote transcription of a gene, for instance. And of course, on the structural biology side, the nucleosome as the kind of smallest subunit of chromatin with about 10 nanometer diameter. Now, these two sides are pretty, established, and there's of course that realm that sits in between that we call mesoscale. That is the organization of chromatin into functional units, into domains, and of course a lot on the organization into domains has been derived nowadays from high C techniques, genomics techniques that use, of course, that are based on analyzing large populations and generate interaction probability maps, and of course also A and B compartments of active and inactive chromatin, which pretty nicely fits into that sort of this dichotomy of U and heterochromatin. Of course, loops and the effect of cohesion is a very important contributor to this organization. Now, of course, a lot of research is being done on how, and it's also an interest of ours, how this 3D chromatin organization guide nuclear function and vice versa. So now our take on that is using to do this on a single cell level using quantitative super resolution microscopy. And in that way, sort of contribute to this knowledge and field. Now, super resolution microscopy, of course, has been center of attention for many years, and you have access to many instruments and techniques, and these days, and typically, most people still work, of course, with confocal microscopy, and this is a nice comparison from a not so recent review, but basically looking at the same biological structures, microtubules, actin, focal adhesions, with different techniques. And so here you have basically the claimed resolution, 200 to 300 nanometer for confocal, I'm going to skip over this, but very popular kind of super resolution technique these days is what we call pixel reassignment techniques, which include techniques like Scan or iSim. And they sort of get down to 120 to 150 nanometer. They use deconvolution for that. So something to keep in mind. And then, of course, SIM, and gonna so interference-based classical SIM gets us down to 100 nanometer. And then we have STAT, stimulated emission depletion, and of course, single molecule localization techniques. And I think I should note here they're often, of course, advertised with a very high number, 10 nanometer, which refers to the localization precision. So it's not quite the same as the optical resolution or structural resolution that ensemble techniques give us. So, but we have to keep the label, the density in mind. But if we kind of stick to those ensemble measurement techniques, like pixel reassignment, SIM instead, then if you look more closely into different structures, it's quite interesting to note a couple of things. So, for instance, if you look here, the difference between Scan and SIM is probably more than you would guess from the numbers that these techniques are advertised for. So particularly when it comes to very kind of dense features here, like the network of actin at the edges of cells is distinctively better resolved here. And strikingly, if you look at STAT, while you get very high resolution, isolated features like these kind of isolated microtubules, if they come together and form denser arrangements, then that resolution increase gets a bit lost. And that's something that also applies here for instance, in these kind of more dense actin filament structures. Now, why is that the case? We have to really not kind of focus on these top resolution numbers, but what we do have to do is to look at how contrast is actually transferred by the optical system. So if you look at pixel reassignment techniques, they're very, very much, are very efficient in transferring contrast in those lower spatial frequencies. And I'm talking here from kind of small structures going up to very high structures, so with high spatial frequencies. But they usually have these kind of bell-shaped curve, whereas STAT rather gives you distinctively, should I say, tent-like curve. So you can get out to very high spatial frequencies, and theoretically, there's no limit to it. But there's clearly kind of it's not a linear display, and that is where SIM really is different, because it gives us a fairly straight distribution or transfer of frequencies. And that means that it all depends very much where your noise or your background is and where it cuts, crosses over. So when you have, very low background, STAT will win out, otherwise SIM will win out. So, and it's particularly this area between, the classical diffraction limit and the twofold resolution limit that SIM can achieve, where you get actually very high efficiency of information transfer. So that makes them really, really good for studying the nucleus, because the nucleus is notoriously dense and crowded environment, and it's therefore very challenging for advanced imaging. Now, we use these days mostly an OMX 3D SIM microscope with a very high NA objective, 1.50, and with this we can go, down to kind of a structural resolution. If we look at the frequency distribution, again, very straight line here, and we can basically get down to less than 90 nanometers in the blue emission spectrum. So that's really nice. And if we look at axial resolution here using 100 nanometer beads and checking out the full width half max, 250 nanometers. So that's well in the range of 3D STAT, but with very kind of efficient, much better contrast transfer. Now, of course, you can look at DAPI-stained nuclei, and this is a nice kind of just comparison between wide field plus deconvolution and 3D SIM. And I think it's pretty clear here how much more level of detail you get, which is a lot more than you would imply from a twofold resolution increase. So in this way, we get also fantastic optical sectioning and separation of features along the Z-axis. And for instance, we can resolve here these nice kind of DNA stain free voids of at the sites where actually nuclear pores are sitting, and you can really see now, and this was one of the kind of revealing things when we used this first time, that we actually can see distinctive kind of organization of chromatin, not just a foggy haze and some channels that lead to nuclear pores. And basically, this allowed us to separate, chromatin domain from interchromatin space, and sort of define these two distinct, say, compartments. And in this sense, we can basically look at chromatin in some sort of topographic way, almost like a landscape of mountains and valleys and lakes, if you like. So and of course, one big question is how does this sort of landscape relate to topologically associated domains, and therefore we have to apply FISH. And one thing that we knew already from the past, but basically assessed a bit more systematically, is that classical FISH techniques that are based on heating, heat denaturation, do affect that kind of fine structure, usually on a which you wouldn't really see on a classical conventional microscopy scale, but at super resolution you can really see that what you usually see in terms of distinctive domain and interchromatin space gets somewhat lost. So we collaborated with colleagues in Oxford who at that time figured out an alternative way to do FISH in a non-denaturing way. So that's the classical kind of way to do FISH, and that we basically denature all proteinaceous components of chromatin. And while you don't affect distances maybe, but that sort of complex contextual information gets lost. So this non-denaturing fish method, termed razorfish, uses basically an enzymatic exonuclease 3 digestion after nicking single, one side of the DNA strand after sensitization and UV irradiation, and exonuclease will chop up the DNA and create kind of large areas of single-stranded DNA, which then allows to bind the probe. And in this way, we can basically keep the kind of physical integrity of domains intact, and we keep that sort of nice separation of domains and interchromatin space. Or that. Now, when we fish TADs onto that landscape, then we get actually very nice, distinct kind of signals of blobs, basically that co-align with kind of the same features on DARPy that have the same sort of kind of edge and separation from interchromatin space. So it's basically, if you like, one of these mountains is labeled with one distinct signal. And if we label consecutive domains that have been identified as TADs based on high C maps, then you can see curvilinear paths of domain features, but sometimes domains also build up a larger blob, or sometimes a domain may also kind of divide up into 2 blobs and so forth. So there's a kind of a big kind of cell-to-cell variability, and that, but clearly, we have here a chain of domains, if you like. So one question, of course, that is apparent here is what is then actually in this interchromatin compartment? And to address this, we apparent candidate was, okay, that maybe RNA, so we labeled bulk RNA with the long kind of 1 hour pulse of 5 EU. And then you can see a very similar sort of picture, but essentially an inverted labeling. It's almost like the flip side of a coin. So chromocenters here, like constitutive heterochromatin, excluded from RNA. It's very strong enrichment in areas where the nucleolus is, and then but a distinctive mutual exclusion of similar sized features that, if you measure them, are like 200 to 300 nanometer wide. Oh, okay, I missed here something. But what is also important to recognize is that, of course, most of the RNA that we look at here is not necessarily messenger RNA. Most of it is actually non-coding RNA, or tRNAs and ribosomal RNAs. Right. So along with RNAs, of course, we have lots of RNA binding protein in the nucleus that are often characterized by intrinsically disordered regions. And these are just 2 examples, HNR and PC, or kind of a factor called DIDO, but this doesn't really matter. And if you look at the conventional image, is just a kind of indiscriminate blur, if you like. So not really a lot to see here. If you apply deconvolution, you see a bit more kind of structure in that signal. And with 3D Sim, of course, you can improve that. It can cut through the noise, but we also know from kind of, maybe people that work in the field know that there are certain artifacts associated with very diffuse signals that have very little modulation contrast. And we basically worked on some filtering methods that sort of exclude signals that have not sufficient underlying modulation. And in this way, we can really filter out the sort of real signals from noise. So we call this modulation contrast filtering. And there we can now really see that, for instance, H&RNPC here resides in the very kind of center of this interchromatin space mainly. And this dido protein, for instance, is also kind of exclusively in this interchromatin space, but in closer proximity to the surfaces of these domain features. So don't want to go into the biology in that sense, but basically just making a point that, of course, RNA binding proteins are very much concentrated in this interchromatin space. Now that we have these domains, of course, there's always an argument is that some sort of fixation artifact, and already kind of, we basically can do STIM imaging of live cells. It's a bit, at that time, we had to do some compromises, and you can see that the signal becomes more and more noisy. Of course, there's a bleach correction, but what you can definitely see, there are kind of domain-like features that have some sort of mobility, but also some level of stability. They don't kind of, let's say, kind of merge or at least on a time scale are fairly stable. They wobble around, but don't merge or disappear. Of course, kind of biophysical analysis, like flow field analysis, show that these features, these blobs, move back and forth and have some elastic properties. So essentially, they are like coherently moving and mechanically coupled, almost with viscoelastic properties. So that's what our biophysicists called it. So they're essentially blobs by definition. Now more recently we've kind of improved on that and basically utilize 2D sim, which is faster and allows us to still get very, very nice kind of distinction here of domain features, and very clearly this is a real thing. It's not it has nothing to do with any fixation artifacts or so. Of course, a classic argument, people outside the field is probably, I mean, this is what we heard from reviewers. Okay, sim, all nice and good, but it's still limited in resolution. Everything will look like globs. What can we get some proof for that? How does that look in EM resolution? And during that time, Harald Hess at Genelia has developed his pipeline of very dedicated focused ion beam scanning electron microscopes, FIBSEM, that operate at extreme high resolution. And importantly, Harold and his team use high pressure freezing and freeze substitution. So basically a technology that does avoid chemical fixation and is recognized as the most structural preserving method. And when he shared his data set of HeLa cells here, and so basically we looked at these kinds of images, and this is just one single cross section of a entire 3D stack, and the resolution here is about 4 nanometers, pixel resolution, I should say. the section thickness was 3 nanometers, so essentially isotropic. And there we basically saw the same features that we saw with 3D Sim in even more clarity. So we have these assemblies of dark dots, and kind of, this is the nuclear envelope. Here are sites of nuclear pore complexes. And then these areas of more kind of indifferent kind of light gray areas with single individual isolated black dots. And of course, our assumption was, okay, probably we're looking here at these mesoscale, kind of these domain features that we also see in live cells, but here as assemblies of nucleosomes. And okay, you can look here, it looks like we have here some linkers, maybe between domains. But then, of course, what is this? Is this here euchromatin? Now, I want to make an argument that this is not euchromatin, but that this is actually our interchromatin and protein complexes with RNAs. So here, for instance, you have ribosomes kind of sitting at the outside of the nuclear envelope. In the nucleolus, you have very evenly spaced. So this is the nucleolus, and you have very nicely evenly spaced dots. We know this can't be chromatin, but these are kind of RNPs here. And if we basically take a 3D data set here like this, so basically that gives you a bit of a sense for the kind of the complexity of the data. Here's the nucleolus, right, with RNPs. And this is just in kind of in comparison, our 3D sim data. And we can basically make use of machine learning assisted segmentation and separate black dots that are in groups and black dots that are isolated. And when we do this and do this kind of segmentation, we can basically separate these features here, and then we can kind of simulate the same sort of resolution that we get with SIM. And this is our simulated SIM image based on the 3D PIPSEM data. And this is what we really get with SIM. And essentially, we when we do RNA staining with DAPI, and basically we see the same thing, kind of an indirect correlation that sort of supports that kind of separation in RNA and the DNA compartment. Okay, so there was a lot about this kind of work, and I want to basically shift gear to a slightly different story, but the basic question is whether we can map single molecules, and the answer, with SIM, and the answer is yes, if these molecules are far enough spaced apart. And where we basically push this was in a more recent work with my collaborator Fina Ox, who for this project was interested to characterize and study cohesion complexes, and developed a nice labeling technique for his halo tagging and using JFX dyes. The basic question is, what can we? Well, it's not a question, but what can we see when we label and look at these labelings with super resolution? And well, basically, you get these kind of images of very clear blobs, no, shouldn't say blobs, dots is maybe a better word. So in classic conventional, not resolvable, but here we can nicely resolve them. When Fina looked at those in closer detail, basically what she noted is that we have essentially dim dots and bigger, like kind of slightly wider and brighter dots. And there seem to be kind of these two different classes. And essentially, if you use intensity or volume distribution, you could basically make up two distinct classes. And the question now is, are the small dots, are they potentially monomers of cohesin? Because of course, in this field, there's always discussion about whether cohesin acts as a monomer or dimer or multimer, and essentially here we can see evidence for this kind of different groups, and by diluting the dye and kind of down so that we only see individual kind of small dots that have the same intensity as these dots, we can use these signals to do stepwise photo bleaching and indeed find just single steps here. So basically, these kinds of experiments, we could derive that the small spots here are in fact monomers of cohesin, and whereas the bigger ones are dimers. And kind of looking into into this more closely with other marks, is basically when you image RAD21 and SMC3 with in this case with a spot tag, same slightly, but still with a single molecule attached to it, you can now really nicely see that you have this sort of co-localization of the same kind of size spots at one cohesin molecule, but actually you can even see, not that we made a big point of it, but actually you could even measure the distance between those spots, which is in that range of 20, 30 nanometer. So definitely more than we would, the size of the complex in a stretched out form would imply, and actually kind of confirming a fold over of the cohesin ring, so that the hinge region and the ATP region come very close together. Now, yeah, so essentially, the next question here in this story was, what is the kind of the difference between those two pools of molecules? And I should have said at the outset that basically the question is, can we derive the information about how the two functional of cohesin, namely sister chromatid cohesion and loop extrusion, is coordinated in the cell cycle. And one of the factors that was characterized in this paper to be exclusively associated with sister chromatid cohesion is sororin. And sororin, basically, if we label this here, really comes out as a single monomer. Same thing if we kind of look at kind of background fluorescent spots, they have the same kind of intensity and size as the ones that we see in the nucleus. So here we have really only one class of molecules. And when we look at the co-localization of cohesin and sororin, then we can see that sororin and RAD21, or that sororin exclusively co-localizes with these monomeric cohesin spots. Basically telling us that, or kind of confirming or suggesting that a sister chromatid cohesion is really exerted by monomeric cohesin, and of course the kind of complementary notion would be that loop extrusion is probably associated with those dimers of cohesin. So something to follow up on. But I want to go back to chromatin, because basically we can say that individual spots that we can resolve here are from single molecules. So basically we can use them as, for single molecule biology. And here I show an image from an immunostaining of histone H3K4 trimethyl, so a marker of active promoters. And here you can really nicely see that these signals sit not kind of everywhere, they're not randomly distributed, but very much linked to the kind of outer regions of these domain features, right? So they're not in the interchromatin space, they're not in the inside of domains, but very much here at the outside. And kind of taking this approach and developing some segmentation, quantitative segmentation of chromatin, we basically could quantify and the mapping of a whole variety of different functional markers of chromatin, kind of from cohesin histone modifications like H3K4, which kind of markers of active promoters, H3K27 trimethyl here, marking repressed chromatin and so forth. H3K20 trimethyl is, of course, associated with repressed chromatin as well. And if we did this with a whole bunch of different markers, and I'm just putting out some kind of examples here. So we can actually use this segmentation into classes, into 7 arbitrary classes, if you like, where we basically allocate class 1 for interchromatin space, 2 and 3 defines the outside of chromatin, like the surfaces, and then 4, 5, and so on, going further to the inside, and basically can create these heat maps. And then we can see this kind of enrichment of, say, polymerase 2 here and HNRMP at this interchromatin and perichromatin side. H3K4 very much enriched at this very outer surface, but depleted in interchromatin space, as you would expect for a histone mark. And then with repressed chromatin marks, they kind of, locate further to the inside. And what is interesting here, so basically we have these active surfaces, repressed cores. And what is very interesting, if you look at so we've done this with 16 different markers, and you can basically look at the sizes of the the enzyme complexes that are associated with each of these marks. And it turns out that the marks that are really associated with the enzyme complexes associated with marks that are sitting at the outside in these classes 1, 2 are also the biggest ones, whereas the enzyme complexes that are linked to the repressive chromatin, those that are really furthest in the inside of domains, are the smallest. So we basically get here a nice kind of hint towards some sort of size exclusion effect of the associated enzymes. Now, if we, so kind of speaking of cohesion, if we, of course, knock out cohesion and do a high C experiment, as has been done in this paper by Rao et al., of course, you will absolutely wipe out any kind of TED landscape. So TEDs are absolutely wiped out here. If you do the same experiment, so we did the same experiment using the same cell line, auxin degradation after 6 hours. And basically, when you characterize, quantitate your chromatin topography and functional distribution of markers, there is absolutely no difference at all. So you really can see how reproducible marker distribution is, and that sort of gives an indication that actually high C and the domains that are kind of derived from that are not exactly physical representations of kind of a physical domain, but really an epiphenomenon of the averaging of large populations. So essentially, it's the domains will form whatever with or without cohesion, and it's only the effect of cohesion loop extrusion that will basically position domains correctly on the 2D scale. So from these data, we derived this sort of CDIC model, and basically that kind of, you know, in terms of kind of sizes, you know, basically, you know, predicts these kind of assemblies of nucleosomes into physical mesoscale assemblies that where silence promoters would sit in the inside and are kind of hidden away so that transcription factor complexes cannot assemble, whereas all transcription activity and enhancer promoter interactions really happen at the surfaces of these domains. And there's a, you can speculate what RNA and potential phase separation of RNA and RNA binding proteins do to kind of manifest this separation. Essentially what that kind of implies is that instead of this dichotomy of euchromatin and heterochromatin, should rather think of euchromatin being at the kind of locating at the domains of at the surfaces of domains, and the interchromatin as a distinct separated functional compartment for RNA processing, and that this kind of separation is functionally relevant. Right. So essentially, we think that it's the surface to volume ratio that is essentially the differentiator between an active A compartment and an inactive B compartment. And in a way, this kind of separation is kind of yeah, how to say, it's basically you can look at it as like islands where activity is really kind of happening at the beaches, and where you find the ice cream and the uh, and whereas the interior of these islands are pretty much kind of hidden away from any access to the good stuff. So anyway, I'm I'm mindful of the time, and I just want to make some mentions on that, the fact that we've only looked at kind of specific cell cycle, we're basically restricted to G1. But there is, of course, an interest in other cell cycle stages and other cell types. And we do, look at this with EDU pulse labeling, and SIM in the past has helped us to resolve individual replicons. And we can basically look at post-replicative DNA here, and we can basically see that DNA that is just undergoing replication is locally massively decompacted. And when we do our mapping of histone marks onto post-replicative DNA, then we do like globally see, you don't see that many differences, although there's some interesting kind of changes, for instance, in cohesion depletion, but I don't want to go into detail. But if you look at post-replicative chromatin only within these kind of masks, and you can see that suddenly, marks that have been kind of hidden away in the interior of domains become now accessible, and that sort of and that sort of separation, of course, is loosened, and that is actually an indicator that or kind of is a strong indicator of DNA replication being a massive kind of force to break up these domain features, at least for a short time, and provide access to otherwise non-accessible areas. And as a kind of extreme case, where you have lots of replication going on, I want to mention yes cells. And if we look at yes cells, then we do see a lot more dispersed chromatin texture, and these cells are constantly, of course, replicating. So here we do not see that kind of these distinct formations of larger domains. There seem to be smaller domains, but it's definitely a different different topography. And on the other hand, of course, if we look at differentiated cells, quite the opposite. So formation of very large domains. So here, if we look at RNA, very much kind of restricted to these small pockets of interchromatin space, much smaller surface to volume ratio. And essentially what that kind of suggests, I basically call this cotton candy chromatin. This is more like a sponge topography, and this is like a Swiss cheese, essentially. basically what that sort of suggests is that there's kind of a hard wiring of transcriptional programs into chromatin structure, and to basically break up and de-differentiate cells. That is sort of what or this that basically going from this in this direction, sort of the natural way, but going the other way back requires certain kind of breaking up mechanism, and replication is definitely one of the factors. Okay, so with this, I want to end my talk. I had a little part on expansion microscopy, but I think I've spent and finished my time. So I want to thank many collaborators, of course, particularly in this case, the lab of Kim Naismith, Fiona Ox, who's now junior group leader in Copenhagen. And of course, Neil Brockdorf. We have lots of very nice work together that I haven't showed. And others in Oxford, and of course, my team of students. So I've showed some work from my PhD student George, and but a lot of the work that I've shown today was basically by my former PhD student Ezekiel Miron and Boel Olenkam, a master's student. So big shout out here. And with this, I want to kind of finish and take questions if there are. Thank you very much.
Thank you. That was fantastic. There are already a couple questions in the chat. If you want to just maybe read those. Do you see
Yeah,
the
okay.
chat?
I'm going. I see the chat. Yes.
Okay.
Two questions. If cohesion doesn't organize that genome, what is its role, and what does organized chromatin domains observe? Yeah, I see what you say. Yeah. Okay. Okay, the role of cohesion, as it seems, is to ensure that, and of course in conjunction with CTCF, that the domains basically, and that certain regulatory elements and mechanisms end up in the same domain and can interact and like enhancers and promoters interact at the surface of a domain. Is if they would end up at separate domains, that would be less likely. So it's probably a way to enhance certain likelihoods. But basically, the formation of domains is independent of the presence of cohesin. So that is a propensity of nucleosomes to interact based on charges. So of course, acetylation plays a main role here, or the absence of acetylation. So why do you think the system phase separates from DNA and RNA? It's a good question. I mean, this is nothing that we kind of that we've investigated, but of course we know that RNA as a kind of small charged molecule, and the fact that it binds with RNA like proteins that are often characterized by IDRs that are known to phase separate, that might kind of promote that RNA and DNA doesn't mix, if you like, but basically are kind of separated. But I'm not a biophysicist, so I'm for others to look into. It's just, I think what we're looking here is a combination of different mechanisms, of which charge, the binding of large, transcription factor complexes, the production of RNA, all kind of contributes to that sort of separation that we see. Okay, very interesting. Thank you. I was a little confused about the volume assigned to a SMC dimer. If I'm not mistaken, it is on a 1 times 10 to the 7 cubic nanometer, which would correspond to a cube with a linear size of about 200 nanometer. Oh, yes. Yes, so basically, of course, our resolution is not of course not isotropic. So we have like probably in that channel maybe 150 nanometer in XY, but in Z we have still 300 nanometers. So that basically the size, the volume would probably correspond to a size of that. I'm not sure if I'm now answering this, going in the right direction. But if the question was, is 200 nanometer too big? Well, maybe it's because of the anisotropic resolution. So thank you, great talk. Can you comment on the dynamics of chromosome? Does the surface and internal chromosome domain rapidly exchange? Very good question, because that's something that we are just investigate using single molecule tracking. So essentially, what our findings of fixed cells imply is that there isn't a kind of a massive exchange of nucleosomes from the outside to the inside and back. So if a nucleosome is at the outside, it will kind of stay there and not kind of travel back and forth. That will definitely change in replication. And again, that's something that we're currently investigating. Watch that space, I would say. There's definitely an element of stability in there, even though, things are dynamic, certain aspects of it need to be stable. Otherwise, we wouldn't say that in a snapshot, that sort of separation of certain flavors of nucleosomes. Slideshow cohesion knockout does not give a difference in the organization. How long did you wait after the knockout? Yeah, good question. So we waited 6 hours. Basically, we replicated exactly the same experiment. So you also, of course, you cannot wait longer, because otherwise you would, mess up with cells mitosis and so forth. So, but basically we just repeated this experiment, and that's what we saw essentially. And we also, of course, looked at the presence of cohesin, because it was tagged to a green fluorescent protein. So basically, we made sure there is, all the cohesin in the cells that we looked at was really degraded. Right.
That
Okay.
was a great talk. I have one last question, if you don't mind. I know it's probably like 7 o'clock your time, so
Yeah, you realize, yes.
I'm
Thank you.
sorry you've had such a long day. What do you think the functional role of the interchromatin space is?
Well, I think there are two. Well, I mean, as compartmentalization will promote probably certain, you know, make certain reactions more efficient, and of course having a reduced sort of kind of reaction surface of you know, at surfaces will probably might improve, you know, could affect kind of the search mechanism. I mean, I'm hand waving because I know in yes cells, you know, this surface to volume ratio is much larger, but of course that the fact, I mean, that also, you know, is probably one reason why yes cells, or, you know, are, you know, pluripotent, because there you have all the accessibility, and yes, transcription factor complexes do assemble there, and so I think the real kind of functional relevance happens when, you know, along a differentiation pathway and by sort of buffering against reactivation. I think there is where it's really become important.
Do you think that's where a lot of the like non-coding RNA regulatory mechanisms are being executed?
Then it is that, yes, I wouldn't exclude that. I mean, there has often been, I mean, you know, there's always a kind of dispute or kind of the question, why do we need all this this huge amount of non-regulatory non-coding RNA? And there might be just a role in kind of you know, contributing to that sort of you know, as a biophysical regulator, I would say, rather than you know, a very specific role.
Thank you. Sorry. Hopefully you have a nice evening.
Thank you.
Thank you so much. Thank you so much, Lothar. It was a really fantastic talk, and as you could see, there were a lot of questions, very interesting questions from the audience. So a lot of
Thank you, Vadim.
through your work. And just maybe to conclude, I would say that I personally find your work transformative, and it certainly made a very significant impact on the way, you know, we think and I think about chromatin and the genome. So it was really a pleasure to have you.
Thank you so much, Vadim.
Thank you so much.
I appreciate that. Next time I come in person, okay?
That would be fantastic. It would be wonderful. All right. I think with this, we can conclude this really exciting seminar, extremely thought-provoking, stimulating. Thank you so much.
Thank you all. Goodbye.